All right, let's talk about one of the most incredible, most fundamental processes in all of biology. It's this tiny, microscopic dance that is literally keeping you alive right now. We're diving deep into mitosis, the amazing way your cells make more cells.
Have you ever really stopped to think about it? How does your body just know how to fix itself when you get a cut? Or how in the world do we go from a single tiny cell to, well, us? The answer to all of that is the same process, and it's happening inside you millions of times every single second. So why do they have to divide in the first place? Well, it all boils down to a few key reasons that are just absolutely essential for us to exist.
Yeah, our bodies count on this for three huge jobs. First up, growth. You started as one single cell, and mitosis is what turned that one into the trillions that make you who you are today. Then there's repair. Get a paper cut? Mitosis kicks into gear, making new cells to patch things right up. And finally, replacement. Your body is always swapping out old, tired cells for fresh new ones. Mitosis is the engine behind it all.
But here's the thing. A cell can't just split in two whenever it feels like it. Nope. Just like any big event, it takes a whole lot of prep work. And that's all part of something bigger called the cell cycle. This cycle basically has three main parts. The first part, where the cell spends most of its life, is called interphase. Think of it as the getting ready stage. The cell is growing, doing its job, and this is the most important part, making a perfect, exact copy of all of its DNA. Once that's done, it's ready for mitosis, the main show where the nucleus divides. And then, for the grand finale, cytokinesis, where the whole thing finally splits into two.
Okay, so now we get to the star of the show, mitosis itself. This is where all that intense preparation pays off in this beautifully coordinated four-part process. It's pretty amazing to watch.
And to help us keep all the steps straight, biologists came up with a really handy acronym, PMAT. That stands for prophase, metaphase, anaphase, and telophase. Let's use that as our roadmap, shall we?
First up is prophase, the P in PMAT. Just think P for prepare, because that's exactly what the cell is doing here, setting the stage for the big split. So a whole bunch of stuff happens all at once in prophase. All that stringy DNA starts to coil up super tight into those classic X-shaped chromosomes you see in textbooks. At the same time, the wall around the nucleus just dissolves, and these tiny little things called spindle fibers start to form. Think of them like microscopic ropes that are going to be super important in a minute. Next, we've got metaphase, M in PMAT, and the key word here is definitely middle. This part is all about organization, lining everything up perfectly. And just look at how organized this is. All those X-shaped chromosomes get pulled into a perfect single-file line right across the center of the cell. It's kind of like a teacher telling students to line up for a class picture. And as they get into position, those spindle fibers we just talked about reach out and latch onto the very center of each and every chromosome. Okay, after metaphase, we hit anaphase, A in PMAT. And if the last phase was about getting organized, this one is all about action. Think A for apart. This is where those spindle fibers really go to work. They start to shorten and contract, and they pull the chromosomes apart. The X literally splits down the middle into two identical halves, and these halves get dragged to opposite ends of the cell. This right here is maybe the most important step, because it makes sure that each new cell is going to get a complete, perfect set of genetic instructions. We are on the home stretch now with telophase, T in PMAT. You can think of this one as the two nuclei phase, because that's exactly what the cell's about to have. So at each end of the cell, where the two sets of chromosomes are now clustered, a brand new nuclear membrane forms around them. The chromosomes themselves can finally relax and unwind back into their normal, stringy state. So for just a moment, you've got this really weird-looking cell. It's still one unit, but it has two complete, separate nuclei inside. The division of all the genetic material is officially done. But hold on, we're not quite finished. We've got one cell with two command centers. The very last step is to divide the rest of the cell, the cytoplasm, the membrane, all of it. And that process has its own name, cytokinesis. This is the final physical split. The cell's outer membrane starts to pinch inward, right down the middle, squeezing the cytoplasm and everything else inside. It keeps pinching and pinching and pinching until, pop, we have two totally separate new cells. And the result is, well, it's remarkable.
From that one original parent cell, we now have two brand new daughter cells. And these aren't just similar, they are genetically identical clones. Each one is a perfect copy of the cell that came before it, all ready to grow up and, eventually, divide all over again.
So let's zoom back out for a second and just appreciate the big picture here. This tiny, elegant process that's happening on a microscopic level, it's truly the engine that drives life itself. Take a look at that number, 2 trillion. That's a rough estimate of how many cells your body is made of. And it all started from one. To keep you going right now, your body's doing this millions of times every single second. The scale of it all is it's staggering.
You know, this quote on the screen really just nails it. Without this constant, super precise division, you wouldn't be able to grow. You couldn't heal from a simple cut. You literally wouldn't be able to function. It is the very thing that keeps life moving forward, one cell at a time.
The accuracy of mitosis is absolutely astounding. But it's a biological process. It's not always 100% perfect. Sometimes mistakes can happen. And that brings up a really critical question. With millions and millions of these divisions happening every single second, what happens when this carefully controlled process goes off the rails? Now that is something to think about. Thanks for joining me for this explainer.